To effectively fight the wide range of pathogens, the lymphocytes of the adaptive immune system have evolved to recognize a great variety of different antigens from bacteria, viruses, and other disease-causing organisms. On B cells, these proteins are the immunoglobulins, Ig, which these cells produce in a vast range of antigen specificities. Membrane-bound form of immunoglobulin on the B cell surface serves as the cell's receptor for antigen and is known as the B cell receptor, BCR. And a secreted form of immunoglobulin of the same antigen specificity is the antibody. Produced by terminally differentiated B cells, plasmablasts, and plasma cells. Antibodies is a large, Y-shaped protein used by the immune system to identify and neutralize a unique molecule of the pathogen, called an antigen. An antibody unit consists of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains connected by disulfide bonds. Light chains consist of 1 variable domain VL and 1 constant domain CL, while heavy chains contain 1 variable domain VH and 3 to 4 constant domains CH1, CH2, CH3. Structurally, an antibody is also partitioned into 2 antigen-binding fragments, Fab, containing 1 VL, VH, CL, and CH1 domain each, as well as the crystallizable fragment, FC, forming the trunk of the Y shape. Every B cell begins by expressing IgM as its cell receptor. The first antibody produced in immune response is always IgM, or known as B1 B cell. In isotype switching, immune response may express IgG, IgA, or IgE, or known as B2 B cell. Class switching is stimulated by cytokines, released by T cells, or mitogenic signal from pathogen. The immunoglobulin isotypes are encoded by a cluster of immunoglobulin heavy chain C region genes. The general structure of the main immunoglobulin isotypes, above in upper panel, is indicated, with each rectangle denoting an immunoglobulin domain. These isotypes are encoded by separate heavy chain C region genes, arranged in a cluster in both mouse and human, lower panel. The constant region of the heavy chain for each isotype is indicated by the same color as the C region gene segment that encodes it. The characteristics of antibody isotypes is determined by 1. The number and location of interchain disulfide bonds. 2. The number of attached oligosaccharide moieties. 3. The length of hinge region. 4. The number of C domain. The IgM and IgA molecules can form multimers. IgM and IgA are usually synthesized as multimers in association with an additional polypeptide chain, the J chain. In dimeric IgA, left panel, the monomers have disulfide bonds to the J chain, as well as to each other. In pentameric IgM, right panel, the monomers are cross-linked by disulfide bonds to each other and to the J chain. Antibody can participate in host defense in 3 main ways. First, antibodies binding to and neutralizing a bacterial toxin, thus preventing it from interacting with host cells and causing pathology. Unbound toxin can react with receptors on the host cell, whereas the toxin-antibody complex cannot. Antibodies also neutralize complete virus particles and bacterial cells by binding and inactivating them. The antigen-antibody complex is eventually scavenged and degraded by macrophages. Second, antibodies coating an antigen render it recognizable as foreign by phagocytes, macrophages, and neutrophils, which then ingest and destroy it. This is called opsonization. Antibody first binds to antigens, red, on the bacterial cell through the variable regions. Then the antibody's Fc region binds to Fc receptors, yellow, expressed by macrophages and other phagocytes, facilitating phagocytosis. Third, activation of the complement system by antibodies coating a bacterial cell. Bound antibodies form a platform that activates the first protein in the complement system, which deposits complement proteins, blue, on the surface of the bacterium. This can lead in some cases to formation of a pore that lyses the bacterium directly. More generally, Complement proteins on the bacterium can be recognized by complement receptors on phagocytes. This stimulates the phagocytes to ingest and destroy the bacterium. Thus, antibodies target pathogens and their toxic products for disposal by phagocytes.